At first glance, the soil beneath our feet seems like nothing more than dirt. But take a closer look, and you'll find it's teeming with all sorts of tiny living things. And for those who take the time, studying the wildlife that lives in the soil, including the things that live down beneath its surface, can reveal a whole new world.
We had to just keep going and going and going, and at some point, the hole was way deeper than the height of a person, and we had to do things that I don't normally have to do, like haul the dirt out with a bucket, because it's too far to like throw it with a shovel anymore.
I'm Scott Solomon. In this episode, we're exploring the underground life of a unique part of Brazil with Smithsonian biologist Dr. Ted Schultz. Brazil is famous for many things: beautiful beaches, competitive soccer teams, and lavish carnival celebrations. It's also home to a large part of the Amazon, the largest rainforest in the world. Along Brazil's eastern shore, there's the Atlantic coast forest, home to a rich diversity of species. But in between these two rainforests lies another ecosystem, one found nowhere else on Earth. It's called the Cerrado. Unlike the rainforests to its east and west, the Brazilian Cerrado is a relatively dry ecosystem. It's more like the savannas of East Africa, with grasslands and dry forests. Except the Cerrado is even more biologically diverse than the famed African savannas. It includes more than 11,000 species of plants, 800 species of birds, and 200 species of mammals, and an unknown number of insects. Many of the species that live in the Cerrado are endemic, meaning they're found nowhere else on Earth. After I finished my PhD, I spent 2 years as a postdoctoral researcher, and a big part of my research focused on the biodiversity of the Cerrado. One of my postdoctoral advisors was Dr. Ted Schultz, Curator of Entomology at the Smithsonian's National Museum of Natural History. Ted, it's so great to welcome you to Wild World.
Oh, thanks. It's great to be here.
So, Ted, I was thinking back, and I think that one of the first times that we met was during an expedition to another part of South America, to Argentina, that for me was part of my time as a graduate student. Do you remember that trip at all?
I definitely remember that trip. It was one of only, I think, 2 times that I've been to Argentina.
Yeah, I mean, for me, it was a great trip, great experience, and fun to get to be in the field with you for the first time. One of the things that I distinctly recall was at one point we stopped at like some sort of a gas station, and we were buying snacks. And I remember that you discovered the Kinder eggs. I don't know if you remember this. These are those chocolate candies in the shape of an egg, and like there's a little toy inside.
Yeah. I didn't remember that. So that's the first time I had encountered those?
So it seemed like it, because what I remember about that was that, like, once you discovered that each egg has a different toy inside, you immediately decided that you had to collect them all.
Well, that sounds like me.
Exactly. So that was like the moment where I understood that you are a natural collector of things, right? Like, I mean, your job as a museum curator seems like the ideal fit for someone who loves to collect things.
Yeah, it really is. And if you ever visit my home, you'll see that it's just like just massive quantities of stuff that I collect, that my wife collects, but without the organization or the cabinetry that we have at the museum.
So what are some of the other things that you like to collect?
Well, at the top of the list are books. My wife and I are both bibliophiles, and then just paper ephemera in general, and things like trading cards and comic books and pulp magazines and stuff like that. But I also collect objects like and toys, like those toys that are in those Kinder eggs. I have a big collection of Cracker Jack toys and gumball machine toys and all kinds of stuff like that.
That's awesome. Yeah, I mean, So during my time, working at the Smithsonian at the Natural History Museum with you, I always felt like a kid in a candy store. Like every time I remember, getting to go in because, of course, this museum is iconic, and I had been there so many times previously as a visitor, as a tourist, especially when I was a kid and I had a chance to go visit, and I just remember being blown away by how much exists kind of behind the scenes, right? You swipe your ID card and you get to open these magic doors and go back behind the exhibit space. So tell us a little bit about what that collection is like, particularly in the entomology collections where you work.
Yeah, I mean, I never figured it out, but it's probably at least half of the museum is not public exhibit space. It could even be more than that. And for some disciplines that are represented in the museum, like anthropology or paleontology, you might see a whole lot of things out on exhibit. But even so, the bulk of the collections in those disciplines are behind the scenes, and they're never seen by the public. But for entomology, that's just like almost totally the case. We have an insect zoo and a butterfly hall, but there are no preserved specimens in those exhibits. But if you go behind the scenes, of all the departments, entomology has the most individual specimens. And so we take up vast amounts of space. Most of the collections, the entomology collections, are basically mummified insects on pins. That's the traditional way to preserve specimens. But for some groups of insects, it's better to preserve them in vials of alcohol, on slides. So we have, and a number of other very strange ways, like in envelopes for dragonflies and for some Lepidoptera.
Oh yeah, the butterflies with their delicate wings,
Yes.
right? You have to protect the wings.
Yeah. So we have all kinds of collections like that, and we have so many insect specimens. It's hard to determine, but it's probably somewhere upwards of 35 million specimens. Wow. That we can't fit them all in the downtown museum. So we have spillover collections out in Suitland at the Museum Support Center. Mm-hmm. We have collections out in Beltsville at the Agricultural Research Station out there. And we have some collections that are off-site at different universities because specialists on those groups have sort of extended loans of those specimens.
And it's one of the things that's amazing is I'm thinking about when you walk into the collection space, to me, it always reminds me of a library. It looks like being in the stacks of a library, right? Except that instead of books on the shelves, you have drawers typically of these preserved insects, as you were describing. And I mean, it's kind of amazing when you think about how small most insects are. The fact that it takes up that much space really emphasizes the number of specimens, right?
I mean, there are some exceptionally large insects, some beetles, Lepidoptera, but the vast majority are tiny. Like if you look at a pin with one of those insects on it, the pin is way bigger than the insect. You can't, you know, in many cases you have to look twice to see the insect. So yes, that just indicates how phenomenally many specimens we actually have. And it really is like a library. It's basically a reference library of specimens, and it's used like a reference library. So, the major users of the U.S. National Insect Collection are other scientists who either come to visit and spend weeks studying certain portions of the collections that they're interested in, or who borrow specimens. We every day we send out specimen loans all over the world to scientists who need to refer to specimens. And among those specimens are type specimens. We have those are the specimens that are associated with the name of an organism. So if you describe a new insect, one of the things you have to do is designate a type specimen, and that's because you might turn out to be wrong. And someday in the future, someone might look at that specimen and say, oh, that's something that was already described and already had a name. Or they might say, oh, this group of specimens this person described are actually two species, and the type is one of them. And, and this has a lot of practical consequences because every day I work interleaved with colleagues in the USDA, and every day they get what are called urgents that are sent from ports of entry. And a big ship is being held up somewhere with, millions of dollars worth of cargo because an inspector found an insect on board, and until they can identify that insect and determine it's not a pest, they're not going to let that cargo get unloaded. So, this has, obviously it also has huge consequences for agriculture and identifying pests of agriculture.
Yeah, because if those insects are potentially dangerous pests or invasive species, I mean, it could do billions of dollars in damage. I mean, it has real world, real consequences. It's not just academic research, so to speak.
That's right, yes. And frequently when people ask us about, the value of the collections, what they're really asking is the monetary value. And in the past, and I wouldn't be able to produce the figures right now, but we've on occasion done calculations for how much the collections have saved people, the American taxpayer in terms of invasive species, agricultural pests, and it's huge. It's a huge amount.
Yeah. Yeah. So what are some of the other ways that the collections are used? So talk a little bit about some of the work that you do and how you use the collection.
Well, in addition to what I described before, the collections are a record of the distributions of species through time. Our collection has specimens in it that are like 200 years old. And the collection's been around for like 100, it's getting on to be like 150 or 175 years, something like that now. So you can go in there and you can walk to a cabinet, open it up, pull out a drawer, go to a tray that contains pinned specimens of a particular species, and you can see specimens from 100 years ago, 150 years ago, and you can see where they were. So you can see specimens from places where they no longer occur. And the way I use the collection, so I'm very interested in ants and ant evolution, but in particular, fungus farming ants and fungus farming ant evolution. And when I go into the field, I'm frequently looking for ants that I know exist because I found one in the collection. And I know where that ant was collected, I know who collected it, and I know the date that it was collected on because that's all on that tiny little half a postage size stamp label that's on the pin with the specimen. And that's it. And I also know from being familiar with this group of organisms that represents, one of the only times that thing's ever been collected.
Yeah.
So that gives me a target. I can go into the field and, my best bet is to return to that site and then start looking for that thing there. But I think I was on a trip with you where we tried to do that for something. And we get to the site and there's like a gas station and a shopping mall there.
Yeah.
And so then you have to start thinking like, okay, what was this site like back when this thing was collected, and where can I still go to like sort of virgin territory that's like that site? And in that particular case of that thing we were looking for, I think it took me 20 years to finally find it, and it was way over in Paraguay. My memory of our trip was that it was in Brazil and near Brasilia, but I finally, 20 years later, found that species, but I had to go to Paraguay to do it.
Yeah, yeah. So in other words, these specimens and the labels that are associated with them are like clues about what the ecosystem was like in the past, and it may or may not still be like that, right? Exactly. Yeah.
Yes, in fact, our collections, and when I say I don't mean just entomology, all of our collections at the museum and all similar collections all over the world are, slowly, kind of rapidly actually being data-based. And that database is a major tool for understanding climate change over the short term and the longer term, especially when you get into paleontology and stuff like that. So you can easily see the effects humans have had on a particular habitat by looking at specimens from that habitat going back, say, 100 years. You can see that something might have been, one of the things that someone did with our collections was they traced the spread of the Argentine ant. So the earliest collections occurred somewhere in the South. And then over time, there were additional collections from farther off, and they tended to follow rail lines. So that's the kind of thing you can do. Also, people have worked with our port of entry specimens. So I mentioned earlier that port of entry inspectors send specimens to us, to my colleagues in the USDA for identification. And they generally, we retain those specimens. And so at some point, someone had this bright idea of looking at all the port of entry intercepts and asking the question, what things have had the greatest opportunity to invade North America, and then compare that to what things actually have invaded North America. And those two things, like in a Venn diagram, do not overlap very much. It turns out that the things that have been found again and again in port of entry shipments are not necessarily the ones that are good at invading. Mm-hmm. Yeah. So that kind of stuff.
Yeah.
I mean,
gives you practical information that you can use to modify your practices or do things to kind of prevent future invasions. Yeah. This is Wild World. I'm talking with entomologist Ted Schultz about his research on ants at the Smithsonian Natural History Museum. When we come back, we'll hear about how field research expeditions to places like the Brazilian Cerrado contribute to the Smithsonian's ant collection, and what this research can reveal about the remarkable abilities of ants. If you want to see our wild world for yourself, one of the best ways is with Lindblad Expeditions. Discovery is in the Lindblad DNA. They've been exploring the most amazing places on the planet for more than 50 years. They have the most advanced fleet of expedition ships in the world, and their trips create unprecedented opportunities for guests. Visit expeditions.com to see where in our wild world you'd like to explore next. Welcome back. I'm Scott Solomon, and I'm speaking with my friend and mentor, Ted Schultz, about the enormous collection of insect specimens at the Smithsonian Natural History Museum. So part of your job, right, is to add to this collection, right? To collect additional specimens that will become a part of this reference collection for future generations.
Yeah, I mean, in some ways, that's the best part of my job. As part of my job, I spend a lot of time in the field, although we're talking pre-pandemic here. And that's part of my job. So I go into the field. I generally spend a fair amount of time on any given trip, a month to a month and a half, somewhere on that order. And I just collect ants. And I like I was saying earlier, I do targeted collecting, although I also do just, not so much recently, but in the past, mass collecting of ants too. And then I bring all that material back, and I always have the appropriate permits and permissions from the countries that I'm collecting in, both to collect and to export material.
Right.
And much of that gets added to our collections. Some of it goes back to the country of origin, depending on the arrangements we've made.
Sure. Contributing to the collections at at the site where it's being collected. Absolutely.
Yep.
So you mentioned that your research focuses on ants, and as does mine, of course.
Uh-huh.
So for me, I think it was when I was a graduate student that I first kind of became fascinated by ants, and it was kind of by accident in a way, because I was sort of looking for a research project that would allow me to do field work in Central and South America, because that was a part of the world that I was really interested in and had some experience there. So, what was it for you that first got you interested in ants?
Well, I've been trying to reconstruct this recently, actually, and I was first interested in ants as a young kid. And I was able to date my interests because I remember certain books that were influential. Like one was called The Tall Grass Zoo. It was a children's book mainly about insects, but other creatures too that you might be able to find in your backyard or close by. And then the other one was The World of Ants. And I know when those books were published, so I can kind of date it. I was probably around 6 or 7, and I loved to catch animals. I like to catch toads and frogs and insects. So I used to have a lot of fun. And I lived in a place where there were large, formerly agricultural fallow fields. So me and all the kids in the neighborhood ran around and collected insects. Then I would read about insects. I would especially try to figure out, like, what do I feed these insects? Because I try to keep them alive. And when I read about insects, I read about ants, and I realized, like, oh my gosh, they're social. They'll die for each other. There's a queen that lays eggs. There's worker ants that don't lay eggs. I got really fascinated by that. And over a number of years, I tried to dig up ant nests, but I had no guidance. At that time, there was no internet and there were no books really that I knew of that told you what to do, and no adults in my life that knew anything about it. So I kind of got discouraged, and then I got interested in lots of other things, and now we're talking about maybe age, high school age. So then for years, I pursued other interests, but I was always whenever I could, read about ants and social insects. So I maintained an interest in it, a fascination with it. And in the early 70s, Edward O. Wilson's book came out, The Insect Societies.
Sure.
And I remember reading that book while riding the streetcar and the bus back and forth to this job that I had as a printer in the San Francisco Bay Area. I read that was my reading on the bus, and it really impressed me. And then it still took a few more years, but I thought, I wonder if I could study that, like, as a career, And I went back and took a Bio 101 class at Sonoma State University, and I was just like hooked. And I took an entomology course at Berkeley, and where you had to make collections. Mm-hmm. And it was like I was a kid again.
Yeah.
Catching insects. It was so cool. And then I thought, like, I'm going to apply to grad school for this, Yeah. And so I got back into being interested in ants kind of like in my early to mid 30s.
Mm-hmm. Mm-hmm. Yeah, it's interesting. I'm always fascinated by kind of people's stories and how they get to where they are. And yeah, I mean, for you, it wasn't just a straight path, right, from your childhood interest into. So, what were some of the other things that you did in the meantime? What were you doing when you were, out of college and
well, first of all, like, I kept going in and out of undergraduate education for between 7 and 8 years before I finally got an undergrad degree. And that was at the end when I was pretty clear about my trajectory. And I majored in things like psychology, art history, fine art, then computer science for a couple of years at Berkeley. And then meanwhile, in order to make a living, I worked at just a wide variety of crazy odd jobs, like cab driver, newspaper truck driver. I was a printer for a number of years, like temp agency worker, bartender, lots of stuff like that, like a lot of things like that. In fact, the last page of my CV has a run-on paragraph that lists all the jobs that I can remember ever having done. So, and I had a lot of interest, like I was saying, I collect books and pulp magazines, and I shouldn't neglect to say that for a number of years I also worked as a writer and editor. So I found my way into the Point Foundation, which publishes the Whole Earth Catalog, and at that time, and they published magazines. One of the magazines was called Coevolution Quarterly. It was renamed Whole Earth Review at some point. They published a computer magazine for a while. So I wound up there and worked my way up from proofreader to copy editor and then managing editor. And then I edited an issue of the magazine, and then I edited a book for them. So I also did stuff like that.
Mm-hmm. Now, I know your real passion and your real research focus has been on this one particular group of ants that you were talking about, the fungus farming ants. What is it about that particular group of ants that you find so interesting?
Well, even when I was a kid and I read about all these behaviors, in that book I referred to the world of ants that influenced me at like age 7. It talks about different things like army ants, and one of the sections that goes on for like 2 pages of kid book, large size text, is about fungus farming in ants. And so that always stuck in what I always was fascinated that insects, which have small brains, can do in aggregate, like if they're social, can have very complicated behaviors.
Yeah, like one ant by itself can't do all that much, but when they work together, they accomplish these amazing feats.
Yes, yeah. And that's always fascinated me. And as you know, ants do a lot of complicated things. But I've always thought like agriculture, that really tops them all in my mind. It's like even at some point when I was decided I would go to grad school, and in grad school I was deciding on a project, I thought, if I can just answer one simple-minded question, I'll be happy. And it's, how could something like that have evolved? How could a complex behavior like agriculture have evolved in ants? And that's basically, that simple question, is still the question I'm asking.
Yeah, I mean, because it's something that we have sort of always been taught is a uniquely human thing,
Yes.
right? Like we thought we invented agriculture. I mean, we celebrate the agricultural revolution, like 10,000 years ago, as one of the most significant accomplishments in human history, and it was. And yet here is this group of seemingly simple organisms, these ants, that not only also do it, but they did it for the first time, some 50 million years ago, right?
Right. It's actually, it's more like 66 million years ago now. It's looking like it was coincident with the end of Cretaceous event.
Wow. So that's really fascinating, right? Because here 66 million years ago, as many people know now, is the date for when the asteroid hit the Earth that caused the extinction of the dinosaurs and so many other things. So you think that it's not a coincidence that is also the approximate date for when ants started to grow fungus?
I'm working on something right now, and I currently have very good dates on the ant phylogeny, the ant evolutionary tree, and the fungal, and for the first time ever, the fungal phylogeny. And they match up in that regard. And they match up with that end of Cretaceous 66 million years ago origin.
Wow.
And if you think about it, it sort of makes sense, because following that bolide hitting the Earth, there was a kind of nuclear winter that went on for, not hundreds of thousands of years. But then the recovery from that took place over a period of 6 million years, 7 million years, something like that. And the South American forest was restructured dramatically. But if you're a photosynthesizing plant, that was bad news for you.
Yeah, because it was blocking out all the light of the sun, right? I mean, like all this stuff in the atmosphere would have made it impossible for most plants to grow.
Right. It shut down photosynthesis for, I don't remember, on the order of hundreds of thousands of years. If you're an herbivore that depended on plants, that was bad news for you too.
Yeah.
But if you're a detritivore that depends on rotting plants or decaying plants or anything like that, it's probably not so bad. It might actually be kind of good for you.
Yeah, there's plenty of stuff decaying, I'm assuming, after such a devastating event.
Yes. And if you're an ant that's already in association with a fungus like that, and I think they were in a non-agricultural association with fungi.
Meaning that they were eating fungi, they just weren't like growing them themselves. So more like a hunter-gatherer.
And they may have been in a sort of the fungi may have been commensals in their nest and stuff like that.
So just kind of there, but not necessarily doing anything helpful or harmful.
And probably not being cultivated. Mm-hmm. But that one of the things I've gotten interested in as a corollary to what I do is the history of human agriculture and other groups of organisms that practice agriculture. And it turns out it's not uncommon.
Yeah. So we know of some other insects like termites that grow fungi in a form of agriculture, right? What else?
Ambrosia beetles, multiple origins of fungus cultivation in ambrosia beetles. And then if you're not just talking about cultivation of fungi, but of other organisms, there are fish that cultivate algae. There's a fair array of things that practice either full-blown agriculture or what you might think of as proto-agriculture. And it's also the case that humans, a large part of agricultural evolution in humans was sort of pre-conscious thought, like just associations with various plants that happen to be good at occupying human disturbed habitats, or plants that humans foraged on and then brought back the seeds and threw things away in pits or middens, and those plants kind of became commensals. So it's fascinating. It's really fascinating.
You're listening to Wild World. I'm Scott Solomon, and my guest is my former research advisor, Ted Schultz. When we come back, we'll talk about some of the extreme measures Ted and I have had to take to study fungus farming ants in the Brazilian savanna known as the Cerrado. The Rice University Traveling Owls program offers exciting intellectual itineraries to destinations around the globe. Traveling Owls trips serve as a catalyst for lifelong learning and strengthen bonds between Rice University alumni and friends. You don't have to be a Rice alum to participate in Traveling Owls programs. Visit alumni.rice.edu/travelingowls to see a list of upcoming trips. This is Wild World. I'm your host, Scott Solomon, and I'm speaking with Ted Schultz, Curator of Entomology at the Smithsonian Natural History Museum. Ted was my postdoctoral research advisor, and he taught me so much about my favorite kind of organism, the ants. And so this kind of work is what has brought you and me down to South America, and specifically to this amazing Cerrado ecosystem in Brazil, where you've been going for many years now, right, to try to find some of these ants, and particularly some of the species that are not really as well known as the leafcutters, right?
Oh, yeah. The leaf cutters are very conspicuous, and everybody, all humans know, well, all humans that interact with them know what leaf cutters are because their nests are, I mean, they're just amazing. Their nests are huge. They're very populous, you know. Some species can have like 6 million individuals in a nest. But they're, as I said, they're a recent development in fungus farming ants, and there's all these other groups that are very inconspicuous, and you wouldn't notice them unless you were looking for them. And and a lot of them occur in the Cerrado.
And so you're reminding me, talking about the inconspicuous nests, of another of the early field trips that you and I did together, where we were looking for the nest of this one particular species of ant, Mycetogroicus cerradensis, right? Which at the time nobody had ever really seen their nests, or at least, scientifically described what the nest was like. And we didn't really know much about their biology.
Oh, yeah. I mean, this is the problem with entomology in general. Just because you know an organism exists and just because you have specimens of it doesn't mean you know anything about the biology. And yeah, we knew they existed because someone had collected some specimens, and because of that, we knew at least one of the places where they occurred, But other than that, we knew nothing. Yeah. That's the trip where we had to dig like crazy, right?
That's right. What I remember about that trip is, well, first of all, I think it took us several days at this one field station. We're in, sort of central Brazil, in the state of Minas Gerais, in kind of the heart of this Cerrado ecosystem.
Yeah.
And we knew that people had collected that ant species from there, but it took us like multiple days just to be able to find any nests, right? I mean, It's not like the leafcutter ant nest can be, 15 feet across or something like that.
Right.
And we learned that this species has a nest that, if you're lucky, you might find a hole with a couple of grains of soil around it. But if it's rained recently, then you're not even going to see that.
Yep.
And I don't remember how we found them. Maybe sometimes we put like something out as bait, like some little grains of
that's
polenta or
what
something.
I usually use is cream of rice.
There you go.
Because it's white, it shows up against a dark background, and a lot of ants will take it. A lot of fungus farming ants will take it, and other ants too. So sometimes you're chasing an ant, and then you realize, like, oh, that's not my target organism there. But yeah, and so a lot of these so-called lower fungus farming ants, unlike the leafcutters, they don't forage in large columns. Like, they might just forage individually. Like you say, their nest entrances are totally inconspicuous, so they're not easy to find.
No, not at all. And so you have to just kind of get lucky and find one ant carrying one little grain of what was it, cream of wheat or whatever, that's like carrying it back, and then it disappears somewhere, and you're like, oh, there must be an entrance to the nest here. I think we found, if I'm not mistaken, 4 nests total on that trip, and we just decided, okay, we're going to try to excavate all of these because we wanted to not only kind of understand what the structure, the architecture of the nest is, because each species has its own unique nest architecture, but in order to understand more about its biology, we needed to get a sample of the fungus that it's growing, and nobody had ever seen it before. Nobody had made any collections, so we had to get down into that chamber underground where they're growing their fungus.
Yep.
And do you remember that excavation?
Yes, I do. I think we split up into pairs, and each of us took a nest. And I remember walking over and watching the other two groups, but I mainly remember ours. And, even at that point, I dug up lots of fungus farming ant nests, and I know that the depth varies. So at that same spot, another species of fungus farming ant, I could probably dig it up a foot down.
Yeah.
But in this case, we we were following the tunnel down, and you lose it and you find it again, and we'd kick off at the end of the day, and you'd come back the next day, and the ants would have re-excavated, so you could see where they were coming out of the wall of the thing that you dug, the hole that you dug. And then you just start over again.
And the tunnel is super narrow. I mean, I'm thinking about the width of that tunnel is maybe a centimeter, maybe less than that, right? I mean,
less.
Yeah.
I think less. Yeah. Maybe half a centimeter. Yeah.
So it's easy to lose.
Yeah, and very. Yeah.
And then if you don't know
lose.
where that is, then you have no real hope of finding this chamber, right? Which is small. Also, it's not like the chamber is that big.
Oh, the chambers that we found were maybe 6 inches across, 8 inches across, something like that.
Yeah, it's like the size of a baseball or a softball or something like that.
But the thing about that is like, of all the ants I've ever dug up, those were the deepest.
Yeah.
And we had no way of knowing in advance because no one had ever dug them up before. Other fungus farming ants in the immediate vicinity could have nests of as little as a foot deep or maybe a meter deep, something like that, right? But a meter would be deep. I would consider that pretty deep. Yeah. But we had to just keep going and going and going. And at some point, the hole was way deeper than the height of a person.
Yep.
And we had to do things that I don't normally have to do, like haul the dirt out with a bucket because it's too far to like throw it with a shovel anymore.
That's right. I remember, I think I maybe spent one day just expanding the hole to be able to have a step to get in and out, right? Because at some point, like, it becomes dangerous if like you can't get in or out. And there was certainly a moment for me where I was kind of at the bottom, near the chamber, where I kind of looked up at the kind of dim light from the hole above me, and I was like, I really feel like I'm digging a grave here. It's a bit grim, but eventually we found it, right? And I actually just looked this up again to refresh my memory. It was almost 12 feet underground where we found the fungus chamber. But what a exciting moment, right? I mean, we know that we are the very first people presumably to ever see this particular species of ant growing this fungus.
Yep. I think about that all the time. Yeah, because who else would try to dig up a nest like that? For what reason?
Right. Yeah. And I think it took us like 4 days total time to get to one or maybe two of the nests where we ended up actually getting to the fungus chamber. Yeah.
I have some slides of us standing there probably at the end of those 4 days, like filthy, and holding various digging implements.
Triumphantly.
And even now, a number of years later, I needed a sample of those ants for a kind of genomic sequencing that we're doing that requires kind of good samples. And I realized, stupidly, like we did not have anything preserved well enough for that purpose. So I needed to go and get more of any species from that genus, Mycetogroicus, but there's only 4 species in the genus, and some of them are even more poorly known than that one, Right. So I did some research, and I saw that I could get a different species up the Araguaia River, and I reasoned that it probably wouldn't be as deep because it occurs on the banks of a river.
Oh yeah. So the water table's probably higher there. So
that's what I figured, and we found them. And I did this for convenience, so that we could get genetic samples. And it turns out they're every bit as deep. They're underwater during the rainy season.
Wow.
How they do, all I can tell you is that when we finally got to the bottom chamber, unlike most ant chamber, fungus chambers, it had a lot of little funny blind tunnels going off in all directions. And I don't know if those were somehow to trap air bubbles or something, but I think those ants spend the rainy season basically with their unable to get to the surface and their chambers submerged.
I mean, that just points to how fascinating it is that each of these species has their own architecture. I mean, they build these structures.
Yes.
It's not like they have construction blueprints to follow, right? Or like a general contractor kind of saying, dig more over here, dig less over there. I mean, they somehow know instinctively that this is the structure that they need to make in order to survive in their environment. It's fascinating.
Yeah, it is fascinating. And the variation, like I was saying, at that same site where we dug up those super deep nests, other fungus farming ants, in other genera, can have shallow nests or moderately deep nests. And so that is hardwired into them somehow. And it also must serve some purpose that we don't understand.
This is Wild World. In our final segment, we'll hear more about field research in the Brazilian Cerrado and how museum collections are helping to archive its unique biodiversity. I love to travel and experience new places, and I've had the great pleasure of joining several Rice Traveling Owls trips operated by Lindblad Expeditions. Each of these trips, from the Galapagos Islands to the Belize Barrier Reef, Baja California, and the upper Amazon River, has been absolutely incredible. Lindblad Expeditions make nature and wildlife accessible to anyone. Visit alumni.rice.edu/travelingowls or expeditions.com to learn more and to see where in our wild world you'd like to explore next. Welcome back to Wild World. I'm Scott Solomon, and I'm talking with Smithsonian entomologist Ted Schultz about some of the research we've done together on fungus farming ants in the savanna ecosystem of Brazil, known as the Cerrado. What are some of the other discoveries that you've made in the Cerrado, or some of the other, experiences that you've had kind of looking for this really interesting but not very well-known group of fungus-growing ants in this part of Brazil?
Well, I've had a number of, it's relatively easy to work in unless you have to dig a 12-foot hole or something. But, so I've had a number of great experiences, but at one point we could tell it was a fungus farming ant. There were like 2 specimens or something. And we knew where they'd been collected, one in a pitfall trap near Brasilia, so in Cerrado. And we couldn't exactly place it to any genus in the fungus farming ants, but we could tell it was a fungus farming ant, which is a interesting thing to think about on its own. But so we went to look for it, and it took quite a while, but we ultimately were able to find a couple of nests and dig them up, and we wound up describing it as a new genus, Cyatta, Cyatta.
Mm-hmm.
So that whole experience, that and just, over the years, I came to realize, like, in some places, savanna habitats are like somewhat depauperate, but the Brazilian Cerrado is not one of those. It's got a fairly long history. Something like about 4 to 10 million years old, and it's expanded and contracted over time. But it's been quite enough time for all of these species to become endemic there, plants in particular, but ants and often the closest relatives of these things are not necessarily Cerrado residents, which tells you that, over that sort of 4 to 10 million year time span, things have come in from the surrounding areas and then become native. So the first time I ever went to Cerrado habitat was near Sao Paulo. And it's just, unfortunately, in the state of Sao Paulo, the remnants of the Cerrado are pretty small, most of them. It's very patchy.
Yeah, because there's been so much urban development and agricultural
Yeah.
development. Sure.
It's nearly ideal for agriculture. So I was just reading something, a history of the Cerrado, and as little as, how long ago would this be now? 50, 70 years ago, in the 1960s, most of it was untouched. Once they put in roads and it became possible to do agriculture, it completely changed. So when I was down in that little patch in Sao Paulo, it was the first time there's a fungus farming ant genus Apterostigma. And I had just associated it with wet forests, and a lot of fungus farming ants, especially the so-called lower fungus farming ants, are wet forest dwelling ants. It's pretty clear that the group originated in wet forests. But I was in Cerrado and I was collecting ants and I saw an Apterostigma and that just blew my mind. Like, this does not seem like the right habitat for this creature. A seasonally dry habitat that is not consistently wet. It also seems a little bit not so great for the fungi, because the fungi need humidity. Mm-hmm. And also, if you've ever seen the fruiting bodies, the mushrooms of the ant cultivated fungi, it was probably in a wet forest. And so it's unclear what's going on. It's possible that in the wet period, these fungi are able to fruit. I don't know, but it's really fascinating once you start thinking about this.
Absolutely. And it's the thing that keeps folks like you and me going back to these places year after year, trying to learn more about them. There can be challenges, though, too, of doing this kind of field work, right? I mean, not just the digging the deep pits like we were talking about, but I remember one time, I can't remember if you were with me on this particular expedition, but we were somewhere in the Cerrado trying to do some excavations, and it was so hot that what I remember is, digging in this hole, and we were trying to collect sterile samples of the fungus. So we had our metal forceps that we would use to gather up the fungus, or even just a metal spoon, and we would use a lighter to sterilize that implement before sticking it into the chamber and collecting fungus, so we don't contaminate the sample. And I remember that I was digging, and I had placed the lighter on the surface of the ground just outside of the hole. And I had my head in the hole and I'm digging, and all of a sudden I hear this loud bang. And I pop my head up and the lighter had exploded. It was so hot. The surface of the ground was so hot that the lighter had actually exploded and there were shards of plastic like all around. I was glad my head was in the hole. So at that point, we decided maybe we should do these excavations at night.
Yeah. I've heard our mutual colleague, Jeffrey Sosa Calfo, has told me that story. That I was not along on that trip, but he was.
Yeah. That was pretty crazy. Yeah. And, we should say that none of the work that we've done in the Cerrado or elsewhere in Brazil would be possible without our many collaborators, especially our Brazilian collaborators, right? Researchers like Geraldo Vasconcelos and Mauricio Bacci, Andre Rodriguez, and so many others. Yeah, they're essential team members. One of the things that makes the work that you're doing and that we've done together so important is that not only are we trying to document and understand the species that are living out there in places like the Cerrado, but we're trying to do it before they disappear, right? And
I really feel strongly about that, and part of my feelings is depression when I think about that. Like I was just looking at this book last night to prepare for this. I don't know if you know this book.
The Cerrados of Brazil. Oh, perfect.
Yeah, and estimates. The book's about 20 years old now, and then the estimate was that 80% had been converted to agriculture of the Cerrado. So just think about in the last 20 years.
Wow.
I've heard estimates of more like 90+% now.
Yeah, there's just not that much of it left. And all these species that are endemic to this environment, presumably if all those little remnants that are left, if they all get lost,
yes,
then we are going to lose those species as well.
Which is a depressing thing to think about.
Yeah. But it also emphasizes the importance of the museum collections like the one at the Smithsonian, right? Because it is an archive of, as you said, of what lived there at a particular point in time.
Yes, and that's why when I collect now, I always try to get whole colonies, and I always try to get the fungus too, because I've had the experience of looking at an ant on a pin in a museum collection, knowing when and where that ant was collected, but not knowing anything about its biology, and I would at least like to know what fungus it's growing.
Do you ever think about the specimens that you've introduced into the collection and wonder kind of how people, how researchers, how students, how anyone in the future is going to use it or think about that specimen?
I do. And one of the things I do is I put stuff into cryo storage. So, and I grow up the fungi now and I lyophilize it, and I put that lyophilized fungi, so freeze-dried fungi, into our cryo storage collection. And that means like 500 years from now, even if everything went extinct, which I really hope it does not, researchers could still ask questions about the ants and the fungi and probably other microorganisms associated with the ants or the fungi because of those cryo-stored materials. But my advisor, I once asked him, like, what do you think, my graduate advisor, William L. Brown Jr., what do you think was your greatest contribution to biology? And he thought for like 2 seconds and he said, everything I collected and put into the collections. And over the years, I've thought about that a lot. And I think that's probably the case for me too.
Absolutely. Well, Ted, thanks so much for being with us and sharing your knowledge and experiences about the Brazilian Cerrado. And I hope that we can get out to the Cerrado together again sometime soon.
Yeah, that'd be great.
That's it for this episode. For more information about Ted Schultz's work on fungus farming ants in the Cerrado and other places, check out the link to the Smithsonian's Ant Lab website in the show notes. We'll also have a link to learn more about the Brazilian Cerrado ecosystem. This episode of Wild World was produced by 3Wire Creative. You can follow Wild World on social media using @WildWorldShow, or subscribe using your favorite podcast platform to get notifications about new episodes. Thanks for listening. I'm your host, Scott Solomon. Join me next time as we explore another part of our wild world. On the next episode of Wild World, we'll explore a place where the silence is deafening. You'll want to tread carefully because the creepy crawlies here make it seem like it's always decorated for Halloween. Luckily, my guest is just the guide you want to get you there and back safely, and to learn some important lessons in the process.